Hey Team Bio, welcome to your screencast on linear and cyclic photophosphorylation. I have to start out apologizing for being a rotten teacher because remember when we were naming all of the different names for the light-dependent and the light-independent reactions, we had a ton of different names for the light-independent reactions. We called it the Calvin cycle, or the Calvin-Benson cycle, or the Calvin-Benson-Basson cycle, or the C3 cycle, or the Rubisco-pentose phosphate pathway. But we really only had one name for the light-dependent reactions, and that was the light-dependent reactions or the light reactions. And it turns out I totally forgot that there's actually a third name for the light-dependent reactions, which is photophosphorylation. Okay. What is phosphorylation? This word that's embedded in here, phosphorylation. Phosphorylation is the addition of a phosphate group to a molecule. So, for example, if we take adenosine diphosphate and we add a phosphate group, and this little I just means inorganic phosphate. There's no carbon in here, which is why it's inorganic. It's just phosphate and some oxygens. So if we attach the phosphate group to the ADP, we perform phosphorylation. And we make ATP. If we go the other way, I should have made that arrow longer, sorry. And I think I still can. Sorry for those of you who are drawing this with pencils. If we start with ATP and we break it up into ADP and phosphate, we perform dephosphorylation. So, photophosphorylation is just a fancy way of saying, oh, whoops, these curly braces are not long enough. I forgot the phos part of phosphorylation. Ooh, phosphorylation. The photo part just means doing this with light. Photophosphorylation. Okay, so if we zoom down here, we've looked at this image in class, and you can see here we're attaching a phosphate group to ADP, and we're using the energy of light in order to do this. So another word for the light-dependent reactions, didn't mean to erase that, is photophosphorylation. Okay, and today we're going to be talking about two forms of photophosphorylation, linear and cyclic. Linear you've already seen before. Cyclic is something a little bit new. So I think I gave you this image when you were solving the puzzle, and I like it for a couple reasons. I think it's really detailed and it's easy to see all of the different steps, but I don't like it because it makes you think that the thylakoid membrane is really small and just one little jelly bean. But in fact, you know from the structure of the chloroplast that the thylakoid membrane system is actually super giant and highly folded, very convoluted, as they say. So this is the thylakoid membrane. And it's not a T. And the membrane, this is one continuous membrane all throughout the thylakoid system. Ooh, it's just one giant, highly folded lipid bilayer. And this image makes you think that the thylakoid membrane is just one tiny little jelly bean, when in fact it is many hundreds, if not thousands, of interconnected jelly beans. So imagine we erased this part of the membrane and then we continued the membrane out like this. That would be a more accurate representation of what the thylakoid membrane looks like. Okay, so this image definitely has some inadequate, depiction of what the thylakoid membrane really looks like, but I do really like how detailed it is. So really quick review. Remember, the whole process of the light dependent reactions starts out with a reaction that we call photolysis of water. Photo, light, lysis, meaning to split or break. So we're splitting water with light energy. And in doing so, we create oxygen gas, molecular oxygen, which is going to leave the plant through the stomata. We create a little H+, which is important because this is going to add to the H+ concentration gradient that's building up inside of the TMS, inside the lumen, relative to the outside, called the stroma, not the, what did someone say yesterday? Stromata? Not that. The stroma. Okay, they were confusing stomata and stroma. It's an easy mistake to make. Okay, and then the most important piece that's released during the photolysis of water is electrons, low energy electrons. So we release a low energy electron when we split the water molecule, and that electron is energized into a high energy electron when we gather light that's passed from chlorophyll molecule to chlorophyll molecule through the photosystem, because they can, when light hits them, their electrons get energized, then they fall back down and they re-emit that light energy, but it's not in a random direction. They pass it off in a specific direction until it reaches the center chlorophyll molecules, which for photosystem 2, which is the first photosystem in the chain, I know, very annoying that it's called photosystem 2, the center pair of chlorophyll molecules are called P680. They're called that because this is the wavelength of light that they absorb best at. Okay, so these center chlorophyll molecules collect, because they're not just collecting light energy from the chlorophyll over here, they're also collecting it from the chlorophyll over here, and then these guys, and then this. So as you hopefully are understanding, they're gathering all the light. It's like a giant satellite dish. They're going to focus it. It's going to be focused on the center, and a high energy electron is created out of this whole business. That high energy electron is passed to an electron transport chain where it's going to use some of its energy in order to pump hydrogen across the thylakoid membrane. Remember, this is active transport because relative to the outside, the hydrogen concentration relative to the inside, the hydrogen concentration out here is low, and the hydrogen concentration inside here is high. So this requires energy to pump hydrogen against its concentration into the thylakoid membrane. Where does that energy come from? It comes from the electron that is passed to it, which ultimately got its energy from light. So by the time it gets to the end of this electron transport chain, it is a medium energy electron, and it has to be re-excited in photosystem 1. Photosystem 1 works almost exactly identical to photosystem 2, except there's a different central pair of chlorophyll molecules here. They are called P700. 700. This is the wavelength of light that they absorb best at. Actually, it might be the frequency. Well, there's a relationship between frequency and wavelength. Anyway, this is something, either the frequency or the wavelength of light, I gotta look it up, that these absorb best at. And now, by the time the electron is ready to exit the photosystem, it is a high energy electron again. Now, I said in our last screencast that high energy electrons are very dangerous. They are, because they contain a lot of energy, they can perform chemical reactions. And so just letting a high energy electron loose into the cell is going to be super dangerous for the cell. But the cell needs to take these high energy electrons to a new place. So how does it do that? It packs them up in a high energy electron suitcase. The suitcase is NADP+, and hydrogen. So what happens is that two high energy electrons jump on to NADP plus H+. So this is going to be, right, a +2. So in order to have a neutral molecule here, we have to add two minuses, which is two electrons. So two electrons, which are high energy, jump on to NADPH, and then these go off to the light-dependent reactions, and ultimately these electrons will become the high energy electrons will become part of glucose. So we strip electrons from water and then we put them onto glucose ultimately. Okay, and then additionally, inside of the TMS lumen, we have this hydrogen concentration gradient. So there's a lot of hydrogen in here, H+ ions, which are just protons. So high concentration here relative to here. And, that should be low. And we use that concentration gradient to drive the formation of ATP. This is also going to go to the power the light-dependent reactions. And I meant the light-independent reactions. Did I say light-dependent reactions up here? Yes, I did. That was a mistake. Light-independent reactions. Okay, so both NADPH carrying the high energy electrons and ATP go off to power the light-independent reactions and make glucose. Okay, so that is linear photophosphorylation. It's linear because it's like a chain. We start with stripping the electrons from water, then the electrons are passed all the way down here, and they end up on NADPH, and then they go away. So we need a constant supply of water to keep this reaction going. Okay, this is in contrast to what we call cyclic photophosphorylation, or the cyclic light-dependent reactions. So, in order to start this pathway, we first need to have the same thing happen. H2O has to come in here. It's split up into its three component parts, O2, H+, and also an electron is donated to photosystem 2. This electron is excited with light energy. It becomes, a high energy electron, so this is an HEE, and then it is passed to the first electron transport system, and it pumps, that energy is used to pump hydrogen into the lumen of the thylakoid. Here's where things get different though. The electron, which is now medium energy electron, is given to photosystem 1. Photosystem 1 excites that electron just like it would in linear, but instead of that electron being passed off eventually to NADPH, it is recycled and brought back to this electron transport system, where it's used to pump more hydrogen into the thylakoid lumen. So how is cyclic different from linear? Well, when we perform cyclic photophosphorylation, no NADPH is made. However, we create a wicked H+ gradient inside of the lumen because we are continuing to use this electron to pump hydrogen into this space. And as a result, we can make tons of ATP. Okay, if we're to look at an energy coordinate diagram of this in black, here we see linear photophosphorylation. And if we were to add on to this cyclic in purple, you would see that it starts out the same way. I guess I should make this a little bit bigger. Okay, close enough. Water donates an electron. That electron is energized in P680. It's sent down an electron transport chain. The reason why it's going down here, energy between this point and this point, remember, is because this energy is being used to pump H plus into lumen. Then it would be a medium energy electron here. It's re-excited here, and then it's sent back here. So in purple shows our cyclic photophosphorylation. So as I said before, end result, no NADPH, lots of ATP. So you might be asking, why is it that plants don't just perform cyclic all the time? That's why they're doing photosynthesis. They're transforming electromagnetic energy into chemical energy, and the chemical energy cells use is ATP, and so boom, voila, they've got it made here. And in part, you're right. Cells need ATP in order to function, but ATP is like cash, and it can be spent easily, but it's not easily saved. And so while this is good for a plant who needs to perform a lot of chemical reactions, it's not good for a plant who's trying to grow. Because remember, the function of glucose, this needs to be black. The function of glucose is both to store chemical energy and build the body of a plant. Okay, so cyclic is good for some reasons, and it's not so good for other reasons, because no NADPH is made, the light independent reactions cannot be performed. Okay, so one thing I want to call your attention to, though, is that the cyclic photophosphorylation pathway is good when we have limited water and lots of light energy. So up until this point, we've kind of been talking about photosynthesis in the context of a temperate broadleaf forest, a certain type of ecosystem. But not all ecosystems look like the ecosystem that we live in. So in class next week, we're going to be examining what does photosynthesis look like in a place that has limited water and lots of light. It doesn't look exactly the same as it does in a place with lots of water and sometimes limited light. Okay, that's all.